Hello, welcome to session 4, Invasives Part 2. My name is Caitlin Monroe-Seiler with Saskatchewan Prairie Conservation Action Plan, and I'll be the moderator this afternoon. I'd like to note that this session is being recorded. I'd like to start the session by stating we respectfully acknowledge that we are on the traditional territories of many Indigenous nations and communities, past and present. For millennia, they have worked to protect these landscapes and the life these areas sustain. I would like to thank these original caretakers and acknowledge the ongoing work and presence of Indigenous peoples in Canada today. I am sure you have all figured out by now the question and answer session works, and I don't need to remind everyone that the Feedloop public chat session will not be moderated. So you can just type in the chat pod down there. I'm pleased to introduce our speaker today, Monica Pokorny with the USDA National Resources Conservation Service, and she'll be speaking about managing invasive plants while revegetating native prairie. So with that, I'll turn it over to you, Monica.
All right, thanks a lot, and thanks everyone. I'm glad to be here today. Like Caitlin mentioned, we're going to be talking about different aspects of plant ecology and how they're incorporated into invasive plant management, particularly while we revegetate native prairie. Before we get started, I also want to acknowledge my co-author, Dr. Jane Mangold, who I've had the pleasure to work with for over 20 years on these topics. So many of the projects I'm going to be discussing today, we've worked on together, and a couple of the recent research projects she worked on with her students, and I'm grateful to be sharing the results of that today. Okay, we're going to start out with just a real basic question. What is the goal of invasive plant management? And a lot of times I hear from people or I see in presentations that the goal is to get rid of a weed or an invasive plant. And what I see a lot of times is that they end up with a lot of bare ground. And I really just don't feel like that's where we want to be with invasive plant management. So I try to focus on that the goal is, yes, to get rid of the weed, but we also want to focus on developing desired plant communities that meet some of our land use objectives. And some of those objectives might be that we want to produce forage, or we want to maintain our native species, or we want to improve habitat, or maybe a combination of all of these. But really we want to develop desired plant communities that function at sustainable levels and that resist invasion. So before we get started, I want to just go over some definitions so it's clear what I'm talking about when I refer to different terms. So what I mean by function, I'm referring to some of the basic processes that land needs in order to grow plants. So this is the ability to capture water, capture and cycle nutrients, and capture and cycle energy. And all of these have a way of determining if they're functioning on the landscape when we enter into a revegetation project. For example, if you're looking at water, you might look to see if water is able to infiltrate the soil, or if you have erosion on the site, if you have plant pedestaling or soil crusting, you might have issues with the water cycle that will prohibit any sort of successful revegetation on the site. And similar with energy, we want to make sure a site has good soil texture and topography so that it can capture both water and energy and get that mixed into the soil. We also want to look for living organisms on the site, make sure they're both on the site and in the soil. Is there organic matter on the site? And is it making contact with the soil, which is really important if we're talking about recycling nutrients through a system. And same with nutrients. We want to make sure we have a surface roughness that allows for nutrient capture. And then we're looking at the plant cover, the productivity, and the vigor of the site. And here's just a couple different images at the bottom where you can see this site is a steep slope. It was probably smoothed and rolled prior to revegetation. You might have compacted the soil, and you can see that the water and nutrients, nothing's able to get into the site, it's just running off, versus a site that's well prepared, where you have texture and microsites, and you're able to capture energy and water, nutrients, and germinate plants on a site. So that's what I'm referring to when I'm talking about plant or site function. And then what we mean by structure is usually what we think about with invasive plant management or weed management. And I might be using those terms interchangeably in this presentation, so just bear with me. What I mean by structure then is, do we have undesired plants present? And what is the composition of those plants on the site? Also, we want to focus on what desired plants we have present, and are they able to reproduce on the site? You might be interested in having diversity. So has the diversity been met? Again, what is the composition of the area? And finally, making sure that what we have on site meets our land use objectives. And this is an example of a project where before we could manage the weeds and revegetate the site, we really had to repair degraded processes of the function and the structure. So this is a site outside of Yellowstone National Park that had been an agricultural field for probably 40 years. So it was compacted. There was a hard pan layer, probably 6 inches into the soil. And so plants weren't able to, the roots weren't able to penetrate the site. And so we're left with really shallow rooted species. It's dominated by annual species, annual weeds like cheatgrass and desert alyssum, and it really had no desired species left on the site. So before we were able to manage the weeds and revegetate, we had to go back and address the functioning of the system. We had to rip the soil so that we could have roots that could penetrate deeply into the soil. And then we could start into the herbicide treatments and revegetation treatments on the site. And this is just a photo where we've been at in the last few years with the project, where we're testing small scale testing of different plant species that we're trying to revegetate the site with. Just a few more definitions. We're going to be talking about revegetation during this presentation, and by that I mean that we're returning vegetation to the site. So we're going to have little emphasis on amending ecological function. There might be a couple places where I talk about rehabilitation or repairing a site, and that's where we're starting to address the functioning of a site. But we're not necessarily trying to restore or bring it back to its original state. So a lot of times in these studies and examples, native species aren't necessarily required. And restoration is sort of the high end of these different projects, where we're trying to return a site to its original state and a functioning state. And native species are really required on these sites. So today we're mostly going to be talking about revegetation. In a couple of places I'll be talking about rehabilitation, and I'll try to point that out. And generally I want to point out that as we move from this end, revegetation to restoration, it becomes more difficult to be successful in your project, and it also becomes more expensive. So with weed management, we often end up in a cycle of treating a weed and then having weeds reestablish and having to retreat them. So we might be spraying, we're opening space, which are niches, which if we don't have desired plants present, we end up reestablishing weeds on the site and then retreating them and kind of going in a cycle. So revegetation can be really important in a weed management project to fill that space with a desired species. So revegetation during invasive plant management would be the deliberate introduction of a desired plant or plant species. The goal could also be that you want to speed recovery of the plant community after a disturbance. We might also want to establish desired vegetation that competes with the weeds more readily than maybe other species that are already there, and also introduce any other plants we want to help meet our management objectives. Revegetation is usually done in combination with some of our other tools that we have for weed control, including herbicide, targeted grazing, fire, and biocontrol. One of the first questions we really need to ask ourselves is, with an invasive plant management project, do we really need to revegetate the site? And in the grasslands where we're working in, we tend to have a lot more bare ground, so maybe 20 to 30% bare ground, open space within the bunch grasses. And what we estimate is that if you have between 20 and 30% canopy cover of desired vegetation, you usually don't need to incorporate revegetation, and that your desired plants will reestablish on their own after you've treated the weeds. But when you start getting up into plant communities that have maybe 25 to 50% or more of your weed species, your desired community really becomes impacted to the place where you're losing some of the structure and different functional groups of plants. And so you want to get into more of a long-term restoration, revegetation type project with your invasive plant management. I like to emphasize that when you are entering into using revegetation and invasive plant management, you really think of it as a process. There's a lot of different things to think about that go into a revegetation project to make it successful. The first thing is we want to characterize our site. So by that, I mean, what is the climate like? What is the precipitation like? What time of year are we getting that precipitation? What kind of soils do we have on the site? Maybe even what your aspect is on, if you're on a slope. And those are all really important questions that you're picking appropriate species that are going to be suited to your site conditions. And that'll help you be more successful. The next thing I like to do is to really outline our project goals and objectives. And I do that after looking at the site characteristics, because by knowing what the site is capable of, we then can pick objectives that are going to be more attainable. And that's going to be really important. The next step we take is really looking at what we're going to do for seeding, maybe the timing of seeding, the method of seeding, what equipment we have available. We're going to start looking into our seed mix. And then we're going to think about what we need to do to prepare the site. And site preparation, I would say, is probably the most important step in a revegetation project, because if you have a poorly prepared site, you're going to have a lower chance of success on your site. So it's really important that you, at this stage, control your weeds, prepare the soil, make sure that you have good seed to soil contact in order for the seeds to germinate. And then you're going to implement the treatments and followed up by monitoring and evaluation so that you can do any adaptive management that you need to. And we teach whole, multiple day courses on this, so there's a lot to think about, but I just wanted to point out a couple examples on the slide. So this is a site that might be like a pipeline revegetation project or a roadside revegetation project, and you can see that the soil is textured so that it's able to capture nutrients and water, and they're not able to just move off site. And it's textured in a way that's perpendicular to the fall line of the site. So again, you're able to capture stuff and it's not just eroding and reeling off the site. And then kind of a rule of thumb is if you walk across the site, you want your footprint to be about 1 centimeter deep. You don't want it to be any deeper or your seeds start to get planted too deep and then have trouble germinating. This is an example of a site that was an existing native prairie where weeds were managed, and then we wanted to broadcast seed some new species. And in order to do that, we used an imprinter for site prep so that we're increasing the texture on the site and increasing places where the seed and water and nutrients can get captured and create microsites for germination. So we're talking a lot about some of the basics about revegetation, and there's a lot to consider, and it's really important to think through it because revegetation is expensive, and I would say that it probably doesn't work more often than it does work when you're mixing it with invasive plant management techniques. Also, revegetation has historically relied on a lot of agronomic practices that aren't necessarily appropriate for when you're working in native prairie or an already established plant community. So what we're going to do now is kind of shift into talking about applying plant ecology into invasive plant management, and particularly into practices that include revegetation. And really there's 3 lines of research related to revegetation research and invasive plant management. And we're going to talk about all 3 of those and provide some study examples and then talk about how to implement the results of that into practice. So the first plant ecology concept that we're going to talk about is invasion resistant plant communities. And this really incorporates the idea of niche into plant communities and resource use. So a niche is really what allows species to coexist and creates diversity on a site. So you can have a fundamental niche in an area, which is really the range of conditions where a species is physiologically capable of living. But then there's also the realized niche, which is a range where the species is actually found. I've always thought that thinking about niches is easier and mammal communities or bird communities, because to me, I can see what's going on and it makes a lot more sense than maybe in a plant community. But this is an example of niche separation in a riparian area by birds. So you might have different bird species living in the riparian area, and you might have one species that's a king bird that would hunt insects along the water and over the water. And then you might also have a sparrow in that area that occupies higher areas in the shrubs and it gathers seeds from the ground. And then you might also have a catbird in that area that feeds on insects and fruits and thick brushes. And all of these species can coexist in the same spot because they're basically exploiting resources differently. And plant species also coexist because of niche differentiation or having complementary niches in a plant community. And this has been studied now for some time and probably started back in the 1970s when they looked at how different underground growth space also had different nutrient concentrations. And it's still not really well understood, but what the research implied was that different functional groups of plants evolved to fill selective ecological roles because they had different requirements for nutrient accumulation, nutrient storage, and nutrient concentration within the plant. And some of that different nutrient concentration might reflect a group of plants' life history. So for example, a weedy species or a ruderal species might have a lower nutrient content that might reflect its high growth rate. So foraging by roots is really difficult still to study and understand, but the studies are suggesting that different functional groups of plants might utilize soil nutrients and water from different niches, both in time and space. So this is usually referred to as having different functional groups within a plant community. And what a functional group really just means is that it's a collection of species that has similar traits and for acquiring and using resources within a plant community. So different ways that they use sunlight, water, nutrients, or carbon dioxide. So it might be that they have they occupy a similar niche. They might use the resource at the same time or within the same space in the soil. It might reflect that they have the same growth form or a life history strategy, or even just the same morphology. So a lot of times when we're studying invasive plants and different plant communities and invasion resistance, we break the community down into different functional groups. And that might just be simply that you have different grasses, forbs, shrubs, and trees, or annuals versus perennials, or looking at different root structures within the plant community. The concept then of having diverse communities resist invasion has been studied for a long time. And one of the first people to study it was Elton back in the 1950s. And what he found was that diverse plant communities are really more stable and they are more productive. So they produce more biomass and they utilize the resources more fully, both in time and space. And when you do that, you lower the risk of invasion by a new species. So that was a really important understanding. And then in the last 2 decades, we've probably increased our understanding quite a bit more of this concept, including that species that are similar to each other are more competitive to each other in resisting invasion. And an example of that is this study that came out of California where you had yellow star thistle, which is a late season annual forb. And they found that if you had diverse plant communities that had 4 or more functional groups, those communities were least likely to be invaded by yellow star thistle. But if you had a monoculture of a grass, those communities were the most likely to be invaded, where a monoculture of another desired late season annual forb were the most resistant to invasion. So the important part of this work was that it's important to maintain and establish diverse communities during weed management, but it's also really important to establish and promote species that might be more competitive against the invasive species. I'm going to go over a few different studies now where we looked at this and kind of built on it over time. So this study took place near Bozeman, Montana. And it started in an established plant community, so a native grassland. And we removed different functional groups from the site. So we removed either nothing, spike moss, shallow-rooted forbs, deep-rooted forbs, grasses, all forbs, or everything. And we had 2 different sites. And then we seeded into the sites a perennial invasive plant, invasive forb, which was spotted knapweed. And we looked at the establishment and density of spotted knapweed. And what we found was that when we removed all of the forbs from the site, we had the greatest invasion by the exotic forb. But when we removed all of the grasses from the site, we had significantly less invasion of the exotic forb. And that was a little surprising to us because in these grasslands, the grasses really provide the most biomass and the most cover in these systems. But it appeared that the forbs were more important for resisting invasion. In a similar study to this in Eastern Oregon, it was another removal study where they removed either nothing, annual forbs, perennial forbs, or all of the bunch grasses. And then in this system, seeded in Medusa head, which is an annual invasive grass. And also as part of this study, we looked at how different functional groups were utilizing resources. So they injected 2 different forms of nitrogen into the soil at different timings and at different depths. And what they found was that the bunch grasses and the forbs really differed in the timing and the depth and the chemical composition of the nutrient capture. They did not find that the functional groups used the nitrogen from different pools, but instead the bunch grasses acquired the most nitrogen from all of the pools of nitrogen, and the bunch grasses were the only functional group that really inhibited invasion by the annual grass. So in this study, the key mechanism of invasion resistance really depended on trait variation among the coexisting species and how abundant that species was in the system. Okay, so now let's look at how we incorporate the revegetation into it and invasion resistant into the plant community and our weed management. So this was an experiment that we looked at incorporating revegetation along with our herbicide treatments. So we used a glyphosate treatment. Oh, I should step back and say this is a site in Oregon. It is invaded by Russian knapweed, which is a rhizomatous perennial invasive plant. And we went into the site and applied glyphosate as our chemical treatment. Then we prepared the seed bed. So we're looking at creating safe sites. So we used tilling, burning, or no site preparation. And then we revegetated with 3 different functional groups. So we used a forb, either a bunch grass or a rhizomatous grass. And these were seeded either in monocultures or in combinations of 2 or 3 species. And what we found in this study was that, well, first of all, these are our seeding treatments along the bottom. So we have our control, rhizomatous grasses, bunch grasses, forbs, and then the combination of those. So both grasses, rhizomatous grass and forb, bunch grass and forb, and all of them. And what they found in the study was that when forbs were seeded, they decreased the reinvasion of Russian knapweed, which is this on this side, more than if we seeded just grasses alone. On one of the sites, the Russian knapweed decreased by 50 to 60%. When tilling was added, there was a 35 to 40% reduction in Russian knapweed, and it had the highest establishment of our seeded species. So follow-up management on this site would be really important to enhance the desired species while continuing to suppress the Russian knapweed. So the herbicide that was used was really a short-lived effect because it was just glyphosate. You're just there's no residual, but it was really important to use it in order to get the desired species established, particularly the forb established on the site. So some of the management implications from this work are that when you're managing invasive plants, you really need to focus on maintaining and establishing key functional groups. So when you're maintaining, you might just, through your management, make sure you have an intermediate level of disturbance where if you don't have enough disturbance, sometimes it can get too weedy. If you have too little or too much, it can get weedy. So you have an intermediate level of disturbance that you could like a grazing program. if you're using herbicides, you want to make sure that you're either patch spraying or spot spraying, because it's really important to maintain the diversity that you already have on the site. It's much harder to revegetate and establish the diversity that you have out there. Another thing you can do is just to decrease your herbicide rate or change the timing of the herbicide application, or sometimes even change which herbicide you're using if you want to maintain a specific desired species, particularly a forb species. And then if you're going to be establishing key functional groups through revegetation, make sure your seed mix has a real diverse group of species that fill different niches and would represent different functional groups. Particularly, it's important to have a plant in there, a species in there that mimics invasive plants biology, and then vary the phenology, the growth form, the rooting depth of those species. Okay, we're going to transition now and talk about the second line of revegetation research when incorporated into invasive plant management, and that is the seasonal priority effect. So the seasonal priority effect really addresses how different life histories and contribute to difficulties in establishing plants at a site. So for example, you might have a weedy invasive plant that has a really fast germination rate and a fast growth rate. And you might have a desired plant on the same area that you're trying to establish that has a slow growth rate. It might overwinter as a dormant seed, and so it's going to germinate later than your weedy species. But really, the seasonal priority effect focuses on the idea that species that emerge first gain an advantage over the slower growing species. And this next slide is an example of that. So the emergence of cheatgrass, which is an winter annual invasive grass that emerged in the spring with the seeded blue bunch wheatgrass, which is a lot smaller. This blue bunch wheatgrass is a perennial, native perennial bunch grass in our area and desired plant. And what's really typical revegetation scenario in our region is that we tend to fall dormant seed. So we want to get our desired seed on the ground in the fall. We want it to overwinter as seed and then germinate in the spring. But meanwhile, we have a lot of weedy annual grasses in our area that are winter annuals. So they've come up in the fall and they overwinter as a seedling, so that in the spring we have our desired seeded grass trying to establish, and it's really already starting to compete with our weedy annual grass that kind of gets a head start on growth. So we looked at the idea of the seasonal priority effect in 3 different experiments, a greenhouse experiment and then 2 field experiments. The field experiment, the greenhouse experiment really investigated to what extent allowing a perennial bunch grass to emerge before the annual grass would give it a competitive advantage. So we seeded blue bunch wheatgrass and cheatgrass either at the same time, or we seeded the blue bunch wheatgrass earlier and allowed it to get to the 2 to 4 leaf stage before seeding cheatgrass. And what we found was that when they were seeded at the same time, the cheatgrass had a 10-fold increase in density, and that decreased blue bunch wheatgrass biomass by 57%. But if we seeded the blue bunch earlier, we still saw a 10-fold increase in the cheatgrass density, but the decrease in blue bunch wheatgrass's biomass was only 13%. So the larger blue bunch wheatgrass was able to avoid suppression a little bit more effectively from the cheatgrass. So some of the implications of this work would be that the order of emergence for desired perennials versus exotic annuals is important when you're planning a revegetation project. And that as a manager, we can modify the standard timing of revegetation practices in order to make sure that our perennial species receive a sufficient head start on growth. A couple of the two field studies that we did with this, the first one was a fallow study. So it was seeded into a fallow field, and we seeded blue bunch wheatgrass again and cheatgrass. The blue bunch wheatgrass was seeded either in the fall or during 8 different seeding dates in the spring, where it started in early April and went through the middle of May. And then the cheatgrass was seeded into this field in the fall. So it would be as if it was emerging in the fall as a winter annual. And what we found was that the fall and the early spring, so the earliest spring, like the earliest April seedings, worked well for establishing the perennial grass. It worked better than the late seedings in the spring. At the same time, we found that the fall seeding and the earliest April seedings were the blue bunch wheatgrass plants that were able to suppress and basically get a competitive advantage on the cheatgrass that were growing in the area, and we had less cheatgrass density in the area. So we still had some cheatgrass there, but our blue bunch wheatgrass plants were able to get a head start and have a little bit of suppression on the weedy annual. We did the same study then in native grassland that was already infested with cheatgrass. So, and then we applied the same. We used a herbicide treatment, a glyphosate treatment, either in the fall or in the spring. And then again, we seeded blue bunch wheatgrass either as a fall dormant seeding or in 4 different timings in the spring, starting in early April and going into May. And what we found with this study was that the blue bunch, first of all, let me explain this slide. So these are our different seeding dates. So we seeded either in the fall or early spring to late spring. And then we had either a fall herbicide treatment or spring treatment. And we're looking at the response of our blue bunch wheatgrass cover. And what we found was that blue bunch wheatgrass density and cover was both highest in the spring. So you can see that here compared to our fall dormant seeding. So fall dormant seeding, remember, is what's typical in our region. We also found that if you can apply your herbicide treatment in the spring, we were able to control the cheatgrass better and get our desired species established and up and growing and able to compete more with the cheatgrass in that site. And this is an example of what that site looks like then after the herbicide treatment and getting our desired grasses established. So we know a lot of times in our area, spring applications of herbicide and seeding is just really hard to do because you have spring rains and wet soils and it's hard to get into an area. But what we found was that if we can get into the area to spray the cheatgrass before seeding, it was really important to knock the plant back again and suppress the seed bed. And then also that the blue bunch wheatgrass really did well establishing in the spring, and it also gave it a little bit more advantage for growing for the entire season before the next cohort of cheatgrass started to emerge in the fall. So the seasonal priority effect then provided blue bunch wheatgrass the ability to reduce the competitive effect of cheatgrass. Okay, we're going to switch now and talk about the third line of ecology, applied ecology used in revegetation practices, and that is the successional dynamics theory, which you might have also heard of being called the ecologically based invasive plant management. So the idea of this is that you're addressing invasive plant management in the way that a plant community would normally change, and the factors that cause plant community change. And those are having an available site. So there might be a disturbance on the site that causes an opening of a niche or an available site. Another way plant communities change is species availability, which you can also think of as propagule dispersal, or basically what species are available, what's already on the site, or what could you introduce to the site. And then another way plant communities change is just by individual species performance. So how are they using the resources? How are plants responding to stress? What would be their life history? And all of these causes of plant community change can be addressed through the tools that we have for invasive plant management. And the key is really just to address them in a complementary manner. So I'm going to give you a couple examples now of some studies where we tested this idea in invasive plant management. And the first is in Western Montana, where we had some grasslands that were pasture land for probably the last 40 years. So they were flood irrigated, introduced grasses that had been taken out of production, and the new ownership now wanted to have them for more of a prairie system, a grassland system that would support wildlife and upland birds. And when they removed the irrigation, they were getting a lot of weed species moving in. Like you can see this giant patch of white top, which is a perennial invasive mustard in the site. And we did an experiment on this site where we addressed the 3 different causes of succession. So we used a herbicide treatment to open up the site and create site availability. We used 3 different herbicide treatments. We used 3 different drill or seeding treatment, where we used the same seed mix, but we either drill seeded it or broadcast seeded it to address the species availability on the site. And then we used mowing to address the species performance. And we were hoping to continue to impact the weedy species while we allowed our desired species to establish. And what we found was the herbicide application increased bare ground. And that's not surprising at all. That's what happens in a lot of our projects. And you can see the herbicide worked really well here. But this is where a lot of projects stop. And so if you don't have a desired species on the site, you really need to make sure to introduce it into the site. And what we also found in this study is that when we seeded and we used herbicides, we were able to increase the desired forbs on the site. So we addressed our species availability. We also found that herbicides decreased the perennial weed cover. So we were addressing species performance with that. We did have a lot of lessons learned from this project, and we have worked on the site in the last few years. And I'm sorry that I meant for the photos to show up progressively, but they're not showing up that way. So I'll just explain that in the short term, we found that our mowing really didn't address the species performance enough to continue to use it. So we needed to find another way to impact the weedy species in the longer term. So in the longer term, we decided to start seeding more aggressive desired species. So at first we started with kind of the optimal that we wanted native species on the site, and they just weren't competitive in this site. So we used some non-native wheat grasses. And then we switched to some herbicides that had more residual for better control. And as we've kind of dialed down the seed mix and the site requirements over time, we've been able to establish some pretty nice and diverse plant communities out there incorporating forbs. want to give you a few examples now of some longer-term revegetation projects that address the successional dynamics. A lot of our studies are in weed management tend to be 2 to 3 years, and so we wanted to look at what happened 15 years after some of our seeding projects, where we addressed the 3 causes of succession. And all of these took place in southwestern Montana. They were all bunchgrass dominated native grasslands, and they were invaded by spotted knapweed, which is a perennial invasive plant. So on the site, we addressed site availability with a herbicide. So in this project, we used glyphosate again, so real ephemeral herbicide, ephemeral effect. And then we broadcast seeded. So you can see there's broadcast seeded and different rates, 0 to 12,000 seeds per meter squared. So we're addressing site availability or species availability here. And then we broadcast seeded either intermediate wheatgrass or blue bunch wheatgrass. And then there was also a biocontrol agent. There were actually several biocontrol agents on the site that continued to impact the spotted knapweed on the site. And what we found was that the herbicide treatment was really short-lived. So you can see here, year 2, we still have quite a bit of knapweed biomass. So this is knapweed biomass, and this is our seeded grass biomass. But the herbicide treatment worked well enough that we were able to establish our desired grasses in the 2nd year. And then over time, what we saw was that the seeded grasses were able to increase in biomass from 26 pounds per acre to over 1,700 pounds per acre by year 15. And our weedy species continued to decrease. So at the highest seeding rate, we decreased spotted knapweed biomass by 86% on the site. But I want to point out too that at the 500 seeds per meter squared, which is more of a normal seed rate for broadcast seeding in a prairie system like this, that also did, comparatively well to the highest seeding rate and would probably be more economically practical. And I also want to mention that, on a lot of these projects, we try to be realistic that we're never going to get rid of the weed completely, but at least in this project, by addressing the 3 different causes of succession, we've been able to move it to a more desired plant community and address some of our land use objectives, which on this site is wildlife management. And here's a few photos again. And remember, this is 15 years after we seeded these plots, and you can still see where the plot lines were. So this is a non-seeded area and one of the seeded plots. And again, these were supposed to show up progressively, so I'm sorry if you can't see the whole photo, but you can get an idea here. Again, this is the non-seeded plot. This is a plot that was seeded with intermediate wheatgrass, which is an introduced perennial species, non-native. And this is our native bunchgrass, perennial bunchgrass species. So in a similar study in Western Montana, where we went back and looked at 15 years after seeding, where we looked at the successional dynamics theory again. On this study, we seeded, and you can see the results here, 2 years after seeding, 15 years after seeding. So that addresses the species availability again. We used a herbicide in this case, picloram, which has more soil residual. So the results of the seeding with either no picloram or picloram. And you can see where we seeded blue bunch wheatgrass or intermediate wheatgrass. And in this, we're looking at the response of our seeded grass biomass. So you can see in the first short term, applying the herbicide was really important for getting our desired species established. It was a little less important over the long term, that once they were established, what we found was they continued to really proliferate on the site and expand into other plots and continued to displace the invasive plant, spotted knapweed. And again, on this site, we also had biocontrol agents. So we had multiple different biocontrol insects on the site. But again, the short-term effect of the herbicide was important for opening the site, and then adding the species, addressing our species availability was really important. So some of the management implications then for the successional dynamics theory was that it's really important to address all three causes of succession when you're entering into an invasive plant management project. And we found that it had really desired results in the long term. Revegetation was also needed to restore and maintain certain desired plant communities in the long term. Some of the lessons we've learned is that sometimes we needed a more aggressive desired species to compete with the weed than maybe our optimal native species that we might want to seed on the site. So you can think of that as maybe a bridge between your weedy site and what you might want in the long term, which is a native site, and you're assisting your community to get there. And then on all of these projects, we needed to continue to address the species performance of the weed while trying to promote the species that we want, so we can end up again with our desired plant community. And that is what I have. So if we have time for questions.
Time for a question, so thanks for leaving time for that. I know that's really important to our attendees. Some of the questions are quite lengthy, so I will put them up on the screen, so hopefully everyone can see them. Here we go. So the first one is from Sheila. When you were discussing glyphosate application for control of cheatgrass and then seeding blue bunchgrass immediately following for best success, can you provide a time frame for immediately following? Some herbicides required a waiting period before proceeding with seeding following a herbicide application.
Well, first of all, I would just recommend every herbicide is going to have recommendations for your timing of seeding after application on your label. At least we do in the States. I'm not sure if you have any other requirements in Canada, but for glyphosate, it's a herbicide that once it hits the soil, it binds to the soil and it's not taken up by the plant. And so you can apply, like, we've seeded the day following applying glyphosate, but one of the catches is you really don't want to disturb the soil too much during that process. I think they recommend if you're going to till or anything, you want to wait a couple weeks. But if you're just going in and drill seeding, and in most of the cases here in the Prairie, we're drill seeding in, and so we can do that the next day.
Okay. Along the same lines, do you have any general advice about timing for a spring spray and then a spring seeding?
Yeah, okay, so the spring spraying is going to really depend on what herbicide you're using again, because some of them are contact herbicides. So like a glyphosate, it has to get taken in through the growing plant material. So your plant needs to be growing, and a lot of times the herbicide will need to have a certain temperature too, so over 55 degrees. And so that's again going to vary by herbicide. So you'll want to look at the the herbicide label. And then the spring seeding for up in Alberta, I know here in Montana, up to the Montana border, we recommend seeding before May 15th. So after May 15th, we're really getting too late. You're not going to see as much spring precip, and you're not going to have as much soil moisture. So if you can get in and seed in April, that's better than if you can seed in May. It just gives you that much more spring moisture to get the plants established.
Thanks for that answer. Speaking about seeding, does a seeding rate chart represent one seeding event, or were you seeding these volumes multiple times over the 15 years?
No, it was one seeding event. So
Okay.
this was back, I think they were seeded somewhere in the early 2000s. I'd have to look at the paper, but I would think like 2001. And the reason why we tried using the multiple rates is one idea at the time is that we could kind of flood the system with desired species, but then you also found that there's interspecies composition. So really the 500 seeds per meter squared, which in the States, that would be somewhere, 50 seeds per square foot. That's more of a normal rate if you're broadcast seeding. So, and that seemed to be quite adequate.
Okay. We have another question related to seeding. Can you elaborate on what you mean when you say aggressive desired species? Why weren't those desired species initially used in the seed mix?
Okay, so yeah, that's a great question. What I basically mean is the initial seed mix was meant to be things like Idaho fescue, some of our native forbs, lupin, species that are slower to establish. And we certainly have some native species that establish well. And I guess that's really what I mean by aggressive. We have species like thick spike wheatgrass or blue bunch wheatgrass are natives that really establish well, and we have really good cultivars of those which come up quickly. But what I meant by aggressive desired species are some of our introduced perennial grasses that like intermediate wheatgrass, tall wheatgrass, we know that they just establish so much more easily than our native species do. And that was important in these studies, just to have the competition with the weeds on the site and to still have a plant there that it kind of can mimic some of the characteristics of our native plants and provide a value to the site instead of having just weeds there.
Thanks for that answer. I know there was a couple questions about it. So back, this is from earlier in your presentation. Daniel writes, "Hi, Monica. In the example you gave of the historic agricultural site, did you choose to till primarily because of the hard pan? Do you think it would have been possible to establish desirable species without tilling?"
Yeah, on this site, yes. So this was a site that was flood irrigated and tilled for at least 4 decades, maybe more. And so there was a really hard layer in the soil about 6 inches deep. And the Park Service had tried revegetating this site multiple times in the last decade and without any success. And so in that case, we really had to break up the hard pan. Now, if you didn't want to use tilling, we're incorporating the use of cover crops more these days. You could think of something like that, like seeding in a cover crop for a year or two if you're in a system where you could plant like a radish or a purple top turnip or something that is going to have a better chance of penetrating a hard pan like that.
And there was a question about disc seeding. Are you always disc seeding, or do you use other methods?
Almost never disc seeding. So we're mostly no-till drill. Okay, so minimizing disturbance, no-till drill, especially if it's already an established plant community. Even a lot of the more agronomic sites, we're using a no-till drill. Sometimes broadcast seeding when we have a lot of other desired plants there. And that broadcast seeding never has as good a results as the no-till drill seeding, because the no-till drill allows better seed to soil contact than broadcast seeding. But sometimes when you have a lot of desired species already out there, it's harder to get that equipment across the landscape without it getting bound up with the existing vegetation.
Thanks for that answer. I know we're tight on time, so our next two questions will just be about resources, if that's okay.
Yeah.
Michelle mentioned if you can share some information on the courses available. You've mentioned that earlier in your presentation.
Yeah, and so right now we're teaching a webinar series on planting a seeding. It doesn't address the invasive plant part of it, but it goes through 6 different webinars on planning and doing a seeding, preparing a site, how to calculate a seed mix, what to look for seed quality. And if you go to the Montana NRCS website and you look under plant materials, we have a plant materials TV. It's called Bridger TV and webinar website under plant materials. And you'll be able to watch some of our PowerPoints and presentations on planning a seeding, because we're not doing in-person courses right now, but you can see all of the material, and then in that we're going to direct you to a lot of different resources that are available for selecting plants and creating seeding plans.
That sounds like a very useful resource for probably all of our attendees. Thank you for sharing that. One listener, Rebecca, had mentioned that you had a graphic that showed it was part of a handbook. Is that on that same page, or would this be located somewhere else?
Yeah, so it's a great reference. So the one that was part of a handbook, it was put out by the University of Wyoming and Colorado State University, and it's the handbook for cheatgrass management. So if you just Google that, it should come up, and it's a great resource.
Sounds like it. I think that's all the time that we have today. So thank you so much, Monica, for a really fascinating presentation, for taking the time to answer so many of our questions. I know there's still quite a few down in the public chat box there that we didn't get a chance to, but I know we never have enough time. But thank you, Monica.
Yes, thank you.
And I'd like to call up our next presenter. So Morgan Webb is of SeedCheck, and he'll be talking about seed testing procedures and reading reports of analysis. Morgan, if you're there, you can turn on your mic and webcam. So Morgan, just under my webcam where it says AV Pod is how you can access the webcam and mic. Yes, there we go.
Okay. Little slow, but here I am. So, hello, Caitlin. Hello, everybody. Yeah, so my presentation today is a little bit about when you're accessing seed sources to do reclamation projects, whether you're reclaiming prairie, whether you're doing an oil site, whatever the project is that you're working on. We get a lot of calls from people that aren't used to the lingo, as well as some of the seed testing reports and what they mean. So what I've tried to do is put together a bit of a tour of some of the paperwork that we go through to try to describe a seed lot so that you get best quality seed that you can possibly have, as well as just a little mini tour of what goes on in a seed testing laboratory, so that those seed testing reports hopefully make more sense. I am an accredited seed analyst, a senior seed analyst, and one of the owners of SeedCheck Technologies, seed testing laboratory in Alberta. When we started, just got to figure out how to forward my slides. There we go. When we started SeedCheck, I had been one of those analysts that had probably the strongest expertise on canola more than anything. And when you analyze canola every winter for 12 or 14 hours a day, it gets a little boring. And I had a passion for some of these more difficult to analyze species. And so I'm proud to say it worked out. There was enough business in this specialized field, and Seed Check Technologies is now just about ending its 19th season. So very, very happy about that. We're accredited by the Canadian Food Inspection Agency. We're subject to audits by the Canadian Seed Institute. And our staff are members of the Commercial Seed Analysts Association of Canada. As of yesterday, the seed industry has made some major changes in order to organize itself better and provide better access to seed information and laws and all of the different organizations that are involved in the seed industry. So 4 of those organizations, of course not the Canadian Food Inspection Agency because it's government, have combined. And so you will be seeing a new group that represents us all called Seeds Canada. So that's just a brand new announcement. So that's something that you can look forward to, is a one-stop shop for your seed information needs. So I want to introduce the Seed Check team. I am very privileged to work with these 6 amazing seed technologists, experts in all kinds of areas of seed, from germination to purity to diseases, vigor. Everyone in that picture there has a role in making sure that that we do the best possible seed testing that we can. The combined knowledge there is incredible. Just a few notes: when you become an accredited seed analyst, you usually do 2 to 4 years apprenticeship on the job. You can write different portions and for different crop kinds. So if you're in a corn and soybean lab in Ontario, you don't have to learn all the grasses and that type of thing if you're not going to use that. You can also specialize in just germination or purity when you write your exams. So at least 3 of our staff are qualified in all areas, germination and purity of all crop kinds, including myself. And so that's more of a 4-year apprenticeship. And the exam for that is like 16 hours long. And you have to know over 1,000 species, their scientific name, how to tell them apart, and how to classify them within the Canadian seed system. And so I will be showing you guys just a taster of how we go through all of that. So, okay. Next slide. So what goes on in a seed testing laboratory anyway? Some of the areas that I'm going to cover, or what it means to have a purity test, a germination test, a viability test or TZ. Not going to touch so much on disease and vigor. Those tend to be more for agronomic crops, but they can be important in reclamation species as well. So this is a seed testing certificate. You can see that, I don't know, Caitlin, if my arrow is visible, but in the top left, you can see the laboratory. You can see that we're accredited laboratory by CFIA. That's an important thing to look for. The customer, which would be, if you're sending the seed, that would be you. If you're buying the seed off someone, that would be the seller, or maybe someone else along the way if the seed is transferred different hands.
We can't see, sorry to interrupt, we can't see your pointer. Just
Okay.
Down at the bottom, there's kind of a green square, and to the left of that, there's a pointer.
There's a pointer.
Yeah, it looks like a little arrow. If you click that, you're probably on private pointer. Just change it to, yeah, there we go. You can see it now.
Okay, great.
Sorry, continue on.
There we go. So, and then this section of the report is what the sender supplies to us when the seed lot comes in. And so you can see crop kind, scientific name, which we do our best to try and stay current on, but they do kind of change on us a whole lot. Variety, if that's applicable. Sometimes it's pedigreed seeds, so you'll see this crop certificate number. A little interesting thing about crop certificate numbers, if you do see one, is the first 2 digits are the year that it was grown in. And so you can, if you have a crop certificate number, you can tell the age of the seed. The next 6 digits are specific to a seed grower, so you can actually trace who grew the seed. I know that a lot of times in reclamation species, they're wild harvested, and so this is not going to apply at all, but it just, so you know what this number means. The 8 in front tells me which province it was grown in. The 8 tells me that it was grown in Alberta. A 7 would be Saskatchewan. And then the last 3 digits are just specific to the class of the pedigree. So whether it's foundation, registered, certified, breeder seed, that type of thing, tells me that information. And then the lot number might be specific to that lot of seed. Then you'll see the body of the certificate, this whole chunk down below here. And that's the part where we tend to pack in a lot of information that confuses people. And so I'm not going to go over what that means right now, because I'm going to start a little bit of a tour of the lab first, show you what we do, and then hopefully come back to this and we can look at where we find the information, and hopefully that helps people know more what that means. Okay, so one of the first things that is important when you're sending a seed sample to a seed laboratory is that it is a representative sample that represents the seed lot. And so it's important that these samples are taken with care so that the work that we do does actually represent the seed lot. If it's a larger seed lot, someone will take primary samples from different bags or containers in the seed lot, combine those into a composite sample, and then reduce that down properly in order to get the submitted sample for the laboratory. And triers and cans and hand sampling, depending on how your seed lot flows and what your seed lot is, can be important in different ways of sampling a seed lot. And so, sometimes something, especially if it's a mixture, is really, really difficult to sample, because if you have different size seeds within there. So we could do a whole 2-day course on sampling, but that's just all I'm going to do to touch on it, other than this next slide, which is what some of the sampling techniques that we use in the seed laboratory. So you can see Michelle on the left there is using a riffle divider. Now that divider is designed originally for soil, but it works very well on some types of seed. You can see she's putting a large round rolling seed through that. What is represented there is a field pea, but that would be used for some vetches and that type of thing. It's a divider that doesn't add any additional mechanical damage. Sometimes those hard seeds are very prone to damaging the embryos, especially if they're on the outside of the seed. So it's one of the ones we use if we think the seed lot might be prone to that. And then this divider here is called a gamete divider. It works on centrifugal force. So it actually spins and divides the sample into 2. With both of these types of samplers, we will run the seed through at least 3 times until we reduce it down to the portion that we need for testing. So we don't pinch, grab a spoonful, that type of thing. We let the appropriate methods divider decide the portion that we are going to analyze, so that we don't analyze with bias. But some seed lots, there's more techniques like pouring over little cups, or if they're a mixture of many species, sometimes these mechanical methods are not the most appropriate method. So you can see then we get the sample reduced down to the right portion. There's, we label them all the way along the way so that there's no mix-ups. I have all kinds of double checks in the system when we're testing seed, so that we make sure that the right sample gets the right results at the end of the day. And yeah, it can be very different depending on what crop kind we're analyzing. So. And so one of the most important tests that we do, especially on small seeded crop kinds, is the purity test. And you'll hear a whole bunch of different terminology when it comes to the purity test. So I'm going to try to describe what those mean, and then we will look at where those results are reported on that report of analysis. But normally, in general, the sample is broken down into a couple of portions. One portion is 25,000 seeds, and that is used for noxious weed exam or count of other species by number. And then out of that 25,000 seed portion, we pull out a 2,500 seed portion. And so, so 10% of that sample for what is sometimes called the purity exam or the pure seed exam or the percentage test. And that's when we report contaminants by percent or the amount of pure seed by percent. And I'll show you a little further how that works. And so the steps to this are we take those pre-weighed out portions and we retrieve anything that we find is a contaminant. We identify that contaminant. We classify it according to the Canadian Seeds Act. And then, of course, the final step is to report that. One of the tools that we use, especially on chaffy grasses, which is the bulk of our work, is this seed blower. So seed goes in a cup. It's kind of like a money tunnel. The seed blows up in the air, and you can see there's a tube, a clear glass tube, and light, immature chaffy seeds will blow over into a cup on top, and the heavier seed will remain on the bottom. For some crop kinds, there's actually a designated, and that's what our notes are down here, blowing point for that species. So we calibrate our blower. And so if you send like a side oats grandma to any lab that's doing it properly with a seed blower, you should get the same result. So the idea is to have uniformity amongst labs, because that's one of the crop kinds that has a blowing point. Blue grama does, and then most of the other ones that do would be agronomic crops like orchard grass or turf grasses like blue grasses. So, but we still use it even if there is no blowing point as an aid in separation to break down the sample into the portions that we're analyzing. You can see here is a sample on my desk, and it happened to be a sample of Western porcupine grass. So you can see those guys that will stab into your fingers and your hands when you're working with it. And we break that down into pure seed, other crops, weed seeds, and inert matter. And I'll try to go into a little bit how we decide where to classify those things. And then we weigh each of those portions at the balance into those portions, and then we add it all up and we give you a percentage. So you've got 98% pure seed with 1% other crops and 0.5% weed seeds and 0.5% inert matter. We may also weigh ergot as a separate weighing portion as well. Okay, lost the ability to move my slide forward. There we go. And you can see then we might tuck into the 25,000 seed portion and go through it and pull seeds by number. So we're looking for, noxious weeds, anything that doesn't belong, and other crop kinds in there. We have an extensive seed herbarium in our laboratory, and so we store them in these vials. When we find something in a sample that we don't know what it is, we will depend on these verified samples in order to help us identify something. You can see, here's an example of something that a fairly large seed that we might be struggling with. So cow parsnip, which is a very common species that we find everywhere, probably throughout the country. And then one that you probably all hear about is giant hogweed. And we have to be able to tell those two species apart if we find one, because giant hogweed is on the prohibited noxious list. And many provincial lists as well. So we have to make sure we identify that guy properly. We have collections that are well over 100 years old that we still use to identify. So that's an important part of our work is maintaining a good seed herbarium. If we don't know what something is and we can't figure it out, we send it to the National Seed Herbarium in Saskatoon, which has like well over 30,000 species, and they can hopefully help us out. Sometimes we create our own resources, and so I saw some questions on the previous speaker about cheat. Here's some of the bromes that have been added to the secondary noxious list for Canada. So we've got the cheat, downy brome, Japanese brome, and field brome. So it's important that we have the skills to tell these species apart so that we're identifying them properly for you. And there's an example of something. The 4 seeds here on the left are Kentucky bluegrass, and the 4 seeds on the right are sandbergs or big bluegrass. So it's important that we're able to tell native species of course, especially for your group's purposes, apart from, ones that are considered a crop kind, but maybe are more of an escapee in certain situations. So getting into how we classify contaminants. So you will see us classify things into groups. Weed seeds, other crops, inert matter, and disease bodies. Disease bodies is usually sclerotia bodies, or commonly in grasses would be ergot. And how we seek to do that is we use a few different tools, not just tools, but rules that we are required to follow. The Canada Seeds Act, the seeds regulations, which are the functioning rules under the Canada Seeds Act, the weed seeds order, and the grade tables, which are part of the seeds regulations. And so there's a picture here of Side Oats Grandma. This is one of those guys that probably, especially for the group that I'm talking to today, it's rather annoying when we see a seed lab classify that as a weed seed, but according to the rules, we are forced to put it under weed seeds, even though it may be desirable for you in your seed mixture. So it's important to read the seed certificate to your purpose as much as it is to, just pay attention to the numbers on it. So this is the weed seeds order. You can see that it's broken down into classes. Class 1 is prohibited noxious weed seeds. There are things, this was upgraded in 2016. The prohibited noxious weeds, as well as the other 2 classes, primary noxious and secondary noxious, were populated with native seeds that are found maybe not commonly, but throughout Canada. And so one of the things they did was clean up and take off anything that is a native species because it's just, it's just not productive to call something that might spring up as a native a prohibited noxious that you're not allowed to have in a seed source in Canada, especially for imports. It's, if other countries are saying, why won't you let that in? You already have it. The other thing that was done is they added a lot of prohibited noxious species to the list. And they did that because the weed seeds order for the seeds side was not really in jive with the Plant Protection Act, which had a lot of these other species on it. So they kind of cleaned up that and did, so that's one of the things that they did. And then in class 2, primary noxious, they took away a lot of the annual weeds that don't really persist year after year. They tend to be the ones that come up first, and they pop those over into secondary noxious. And then they added some invasives that people's, in both forage situation and a reclamation situation we're begging for. And then you can see class 4 and class 5 tend to be really specialized for turf grasses, and turf grass mixtures. There's some examples of some prohibited noxious weeds that are probably some of the most likely to be found in Canada. This guy is woolly cupgrass. This is jointed goat grass, dodder, and blackgrass or slender foxtail. And these guys are ones that there have been small outbreaks that in Canada that have been contained. So they're kind of good ones to know because they can come up from seed shipments from the United States, where most of those, but not all, are not on their state seed laws, and so they might be perfectly allowable there on a seed certificate. So something to be aware of is a seed report from a US source may not be tested to Canadian standards. So they may not call these prohibited noxious if they're found in a US laboratory. So that's one of the things, or they may not even mention them at all. So it's important to get a Canadian seed report no matter who the lab is doing, what lab is doing the testing. Okay, and then the other place that we get, other than the weed seeds order, is Schedule 1 to the seed regulations, which is the grade tables. And I'll show you just an example of a few grade tables that I think apply to some of the seed that you might be working with. One of those is Table 11, which you can see. Table 11 is like 3 pages long, and you can see here where it lists out the species that are on Table 11, and some of those are species that are native to parts of Canada, like the western wheatgrass, northern wheatgrass, some of the species, and you will see that the seeds regulations also struggles to keep up with nomenclature to have the current scientific names, but these are the common names also that we are required to use on our seed reports. So you may use thick spike wheatgrass, but we will be calling it something like northern wheatgrass or on a seed testing report. So it's good to kind of be familiar with these tables because these are considered the Canadian common names that we use. Our lab also tries to put the current scientific name on the seed report, so that you can figure out if you're using a different common name than we might be forced to use. And so this is Table 11 continued, and you can see it's broken down into things, categories, or columns. And you can see the first rows are about pedigreed seed. Most species that are sold in Canada, native species or reclamation species, are actually only required to make the minimum standard for the table that they apply to. So that would be the common number 2 column. So they would be allowed 4 primary noxious weeds, 15 secondary plus primary, a total of 100 weeds. And this is per 25 grams because it says that the table is per 25 grams. That's going to be useful when we read that seed testing report. Special columns for brassica species and sweet clover. We're allowed 5%, or if it's not one of these guys, in other kinds, 3% other crop kinds, percent of ergot bodies. And then there's these columns here for pure living seed and pure seed by weight. They don't apply to anything that doesn't fall on the grade tables. So for Blue Grandma, you actually don't need to make the standard on these. So you could have 0% pure living seed and it would still make the standard. If it falls on the table, then you have to figure out which column that it fits in for pure seed and pure living seed. And the footnotes on that table, which are complicated and confusing at times, will let you know that. So it will say, you know, ryegrass needs a 90 pure seed and a 95 pure living seed. You will see there is no germination standard, and the pure living seed is the replacement for a germination standard. And so basically how you figure out pure living seed is you multiply the percent pure seed by the germination and divide by 100, and that gives you the percentage of pure living seed. So if that is how we kind of figure that out for seed testing reports. This table tends to be important for your industry because Table 13, even though it's the forage grass mixture, there is a statement in the Canada Seeds Act that says any species that does not fall on the grade tables in Canada is considered a reclamation species, is to be analyzed according to Table 13. So Table 13 is an important one because we are applying those standards to a lot of reclamation species. And so I just thought I'd list some of the native species that are found on the Canadian grade tables. So that is northern wheatgrass, streambank wheatgrass, slender, western, foul bluegrass, and creeping bentgrass. If we find these guys in your mixture as just a contaminant, we will just list them as other crops. Just about anything else, we will be forced to list under that weed seeds column, which would be a great rule change to the Canada Seeds Act because it's not always functional for all. This slide is kind of out of order, so I'm just go past it. So there's that report again. So you can see now that we analyzed 70 grams of that green needle grass for prohibited noxious, primary noxious, secondary noxious, and other weeds. You can see some of the other weeds that we have listed may be considered desirable to you in this mixture, like the June grass. You can see that the downy brome we have here, we have this odd 0.4. The 0.4, what that means is we've used this 70 grams to analyze the seed, and we found 1 in the whole 70 grams, but we've reduced it down to be the per 25 grams that is found on the grade table. So it's not that we found less than half a weed, we found a whole weed, but we're reporting those numbers as an extrapolated number to represent the 25 grams. You'll see in other crop seeds that some of the other crops are perfectly acceptable in your mixture, and some of them are ones that you consider invasives. So it's important to look at both columns, and you'll see that the seed lab is only required to report those guys according to percentage. And so on that table 13, we would just say there's less than 5% of those. So something we do get a request from people that are doing reclamation is, can you please let me know the numbers in there? Like, are there hundreds or is there just 1? And so that might help you decide whether you're going to use a seed lot. So the other area that we're using in the seed lab to determine the quality of your seed lab is germination. And so when we determine germination, we are determining the emergence and development of those essential structures indicative of the ability to produce a useful mature plant under favorable field conditions. And you'll see us classify seeds into the normal seedlings, that's the germination percentage, abnormal seedlings, dead, fresh, and hard seed. And so abnormal seedlings, a question we get frequently, are seedlings that don't have all their structures, or they're decayed with a fungus or primary infection, and so they are not capable of producing a plant. So a lot of people will say, oh, just add in the abnormal seedlings. That is not something that you should be doing because they are not considered a seedling that will produce a plant. Fresh and hard, however, are a different category. They're a little more complicated. Fresh seeds are seeds that take in moisture, but they won't grow due to some sort of dormant reason. And hard seeds are seeds that don't take in moisture. So we find that more commonly in, purple prairie clover or vetches or those type of species that have a natural seed hardness so that they do germinate over time, which is, a good thing in a lot of reclamation species. So something I wanted to mention in the steps that we do is that we plant from pure seed. So we actually take the seed from that pure seed percentage when this purity analyst is doing a purity, and we plant those seeds in our germination test. And so we have to plant a minimum of 200 seeds for Canadian rules. If you're shipping the seed to the US, we need to do a minimum of 400 seeds. And so that seed that we plant from the purity test is also the number that you multiply by the germination to get that pure living seed. So the fact that it's the same seed actually makes the test more accurate. And then we incubate at the appropriate temperature, and then we evaluate the germination test. And so just a few steps. You can see some of the tools we use. This is Deb, and she is using a vacuum planter to plant some type of clover in this case, may even be an alfalfa seed. And so one thing our lab does is we spread the seed out really far, so we don't get fungal problems from a dead seed affecting your normal seeds. So we do 4 seeds of 50 replicates, quite spread out. And she is just going over making sure there's only 1 seed on every hole. And she'll turn that over, and you can see in the bucket there, this method is called rolled towel. And so she actually folds the towel over and rolls it up and lets them stand upright so that their roots will grow down and their shoots will grow up, so that we know that they're not displaying like negative geotropism, so they're not an abnormal where the roots won't go the right way, which we see with glyphosate damage if someone has sprayed the crop. And then there's another method there. This is Bonnie, and she actually does a lot of disease work in our laboratory, things like fusarium and that type of thing. So we don't introduce fungal problems to our seeding mix. And so she's doing a method that's called on top of paper. So the seedlings are allowed to grow upward without being covered. It's best for seedlings that need light for germination. Another method we use on round rolly seeds, like some vetches, would be this is a forage field pea, I believe, this purple pea. They go on the rolled towel, but we use this little springboard to plant them. And then they're all lined up there, and then it's rolled up as well. And then they go into the incubation temperature that's best for them. A lot of species that don't have a great deal of dormancy will go into 20 degrees that we maintain for plus or minus 2 degrees. So it rarely goes past 20, but it can go up to 22 and go down to 18. But we don't, if it goes below that, then we start a retest. You can see there's like some fungal tests on agar and tests on in rolled towel. A lot of the indigenous species that we do, especially grasses or things that have dormancy, what we actually use is an incubator that's 25 degrees during the day and 15 degrees at night, which is the best temperature for cool season grasses. And then we get to the evaluation point. And so I've chosen a large crop kind here to show you that, we'll evaluate into normal seedlings that have all their structures, abnormal seedlings that might not have the ability to grow into a mature plant, and of course, dead seeds. And then of course, we're recording those results. Here's a pre-COVID seed lab seed. We now are all wearing masks and sitting much further apart than that. And you will see in our lab, so you will see this portion here, that's where we report that 2,500 seed test. This is the 25,000 seed test by number. These are special things on the column, like sweet clover, brassica, and ergot, special columns on the grade tables. This is that pure seed, other crops, weed seed, and inert by percent. So you can see we analyzed 7 grams for that, 70 grams for that, and then the germination would be based on 200 seeds, and we may have germination plus dormant seed equals germ plus dormant. That's important in a lot of things with high dormancy. And how we determine the dormancy is through the tetrazolium viability test. And so you can see in this probably a little bit blurry photograph, trying to take it down a microscope, is some tickle grass. So we peel the tickle grass and we pierce the embryo, and then we let the tetrazolium chloride stain into the embryo, often overnight, but may be different for different species. And this is to make a quick estimate of viability. So really, really dormant species, we might do a TZ test only. The other thing that we might do is do a germination test, determine the number that germinate rapidly in 2 to 30 days, depending on the species. Then we will TZ the remaining seeds at the end of the test to determine whether they're dead or dormant. And that's important in a species like the Agrostis scabra pictured here, because it has a soft caryopsis. So if you push on it, you would think it was a dead seed no matter what. So when they could be perfectly alive. So we prepare the pure seed from purity. Then we do a pre-treatment. That usually involves just soaking to soften the seed. Preparation might be piercing tiny seeds or slicing larger seeds in half so that we can access the embryo. Then we have a staining solution. That might be anywhere from 0.5 to 5% tetrazoleum. A staining time, which could be anywhere from 1 hour to 72 hours, depending on the species. And then a very experienced seed analyst will evaluate the tetrazoleum test. And so here are some sun puffs that we got in from Saskatchewan. And they're very, very difficult to get to germinate. And so they wanted to really know whether they were alive or not. The client did. And so what we did is there is no method for them, but they are most closely related to a thing that is a method for is bougainvillea, which is a plant that we don't suspect to see on the Canadian prairies. So we actually followed that method. So we pre-soaked them between these blue blotters for overnight. And then the next day we sliced them open. You can see the little embryo inside there. And then we stained them at a 1% TZ solution for 4 hours, and we got this lovely pink stain. And so we look for cotyledons and a nice root that are all staining pink. And we also press on each one and check for texture. And it takes a lot of experience for the analyst that does it, but they do start to be really extremely good at it. In agronomic crops without dormancy, when we're doing TZs just for sales, most of our TZ tests are well within a few percent of the actual germination test when it comes off. So that's cool. But on some species, when you can't get a germination test, this is the thing we use to analyze the remaining seeds. This last picture of TZ on these sand puffs is a viable seedling, and then one that is dead that didn't stain at all. And of course, there's all kinds of things in between. But it also is a test that we can use to determine vigor of a seed lot, because the way they stain and their texture can help us decide whether a seed lot is going downhill and it's time to get that planted if we've been holding on to it for a few years. Just another example of a clover of some kind. And so viable, non-viable, you'll see patchy staining. That might not turn into a normal seedling. You'll see roots that are broken. Those seedlings would probably develop without a root. And so that's one of the things we use tetrazoleum testing for. And of course, there's that report again. So when you see that added dormant, or you might see that we did a TZ test right from the beginning, and that is something that we used or that we reported right away. So usually within 24 hours that we get the seed lot in. So for species that fall on the grade tables, you are required to get a germination. The only species where you're allowed to add, in order to grade, you're allowed to add TZ result to germination is Western wheatgrass. But for information purpose, like on this green needlegrass, of course, we need that information to know whether the seed's left or dead or alive. And so that is kind of mostly my presentation. Yes, that was the last slide. So that is kind of what goes on in a seed lab, how we determine those numbers that you might be seeing on a report. Something that I want to say before I forget, you can always phone us. I mean, at our lab, we pride ourselves in being that lab that can do everything, but we have a small team. So usually the person who worked on your sample, who remembers it, or they can pull up, we save all the portions after we're done. So if we need to go and retrieve it and answer a question of yours, we save them for a year. We can usually pull them up and let you know, more information about it. So we definitely encourage questions when you're being confused by that seed testing report. So
thank you so much, Morgan. was a really, really fascinating presentation. We've had a few comments from listeners typing in about how interesting it is. I know when you've spoken at this event in the past, it's always been a favorite of the event. So thank you for taking the time to share your wealth of information with us today. We do have a number of questions available from the audience. So I'll just put a couple up on the screen here so everyone can see it. Sorry. Here's our first one. So how much does a seed analysis cost?
Right, so that's a very good question. So, so a germination may run anywhere in the, just over $20 to $50, depending on the species, because it depends on the degree of difficulty. Purities are the same. They run anywhere between like $35 and $150. So, for example, that Western porcupine grass pure seed that I was showing, a seed analyst that's fully accredited but's never worked with reclamation species before, that might take them 2 or 3 days to analyze. So you can imagine the cost in time goes up. In our lab, we can usually get purities done the same day because we have had so much experience with these species. So, but they're in that, sometimes something that's difficult in that $150 range. And then you add on a TZ, which is about $60. And so, you can be paying a couple $100 for a seed test.
Okay, sounds like it's worth it though. Our next question is from a listener named May. Can your lab do counts and identity of species content in a bulk sample of wild harvest seed that is multiple unknown native species unsorted?
Right, and we certainly can do that. What we might end up doing is just having a conversation with you first, because if we try to do on an official purity test on something that's bulk and hand harvested or field or just wild harvested, and it's just full of everything, it could take us a long time and a lot of expense to do that. But what we could do is a reduced portion and just go through it and let you know what we see immediately in there. And so that would be a lot more affordable way to know where you're starting out, or we can, pull 100 seeds or 200 seeds of what you do have in there, the largest species, and do a TZ on them. Just things to determine whether it's worth moving forward and cleaning up this seed lot and doing more with it. So.
Thanks for that answer. Our next question is a bit longer from Ashley. Do you /seed check have any idea how representative the small sample analyzed is of the actual seed lot? I understand lots can be really large, so I've assumed that weed seeds in particular would be highly variable from those on the certificate, but there's no other way to know.
Right, they certainly can be, and that's where that sampling becomes so important. And that's why when we get a sample in the lab, we follow strict mixing procedures, especially for a purity test, because if you've got tiny seeds, they can settle in a seed lot. So it is kind of important that you take the best possible sample that you can, and then official samples that are sold for final sale of a seed lot are usually after it's processed and cleaned. And so by then the seed lot is usually pretty uniform, even if it's contaminated. And so if you do that sampling properly, it is amazing how it can represent the seed lot extremely well, give or take some variation, right? We have to be a little bit forgiving of, we're dealing with a living thing. And so something with a middle of the road germination or TZ in the 60s and 70s can vary more than something that's really, really nice or really, really bad. And same thing, if something's more contaminated, it can vary more than if it's a fairly, homozygous seed lot. So.
Thanks for that answer. Our next question by Marshall, he's wondering if in order to be found on grade tables, do they need to have varieties?
Right. So when I went through the grade table, you saw that those levels from foundation, registered, certified seed, those are ones that have gone through the pedigreed seed system. And there are some reclamation species that do that. And that means that they've gone through further steps. So an eligible seed grower, part of the Canadian Seed Growers Association of Canada has grown it. They've got their seed from a reliable source like a plant breeder, and they've had a crop inspector go in and check that is true to variety. But a lot of stuff that we're working with in this reclamation area probably most stuff has not been through that. And so we may not know the variety. It might be wild harvested. Or if we do know the variety as common seed, we have to be careful whether we use it or not, because there are laws within Canada. You can use the variety on some species without retribution, without breaking laws, and there are some things you cannot. So that's a whole other area of kind of interest, when you can and cannot use varieties. But usually when something doesn't fall on the grade tables, varieties are acceptable for use. It's when they do fall on that they're not.
Gotcha. There's a comment from a listener named Tracy, and she said it would be great if SeedCheck could offer a webinar on the website about how to read a seed test certificate. I should mention that this webinar here is being recorded, so
Right.
People can refer back to it.
So this webinar is like basically the early overview of that. It would be, it would have been nice if we could have had an hour or two break off if we were in person, especially to hand out certificates to people and have them go over them and look for what might be wrong or what they're worried about or what they should ask questions about. And certainly we don't have anything like that online at this point. It is our absolute busiest time until the seed is planted. And then over the summer, we could think about spending some time and designing something like that. It's a great idea.
Yes. Yeah. It can be quite time consuming to do something like that, I guess. And I imagine you're pretty busy at this time of year. Everyone's getting ready for spring seeding.
We certainly are. We go to like 4 times the amount of work in January, February, and March. So yeah.
Wow.
The team is focusing hard. Yeah. So
we host this event at this time of year because usually people are not quite as busy as they are during
everyone else is not as busy, and then we're slow in the summer, so we get to go out and go on the nature walk. So yeah, sorry
nice.
Guys that are busy in the summer.
Oh man. That's all the questions that we have at the moment. I'm not sure why our group is being so quiet today. Usually they're pretty chatty. To all of our listeners, if you have any more questions, type it in. And there's been quite a few comments that have come up. So while we wait for a few more questions, I just wanted to publish a couple of these comments. So Andrew says, "What incredible skills," and I agree with her.
Some people, when you say you're an accredited seed analyst, they either think it's boring or they think it's the most exciting job ever. And we think it's the most exciting job ever because you're constantly, like what all of you do, always learning. So
yeah. And Rebecca Magnus, she's part of our planning committee for this event. She says, "You made reading seed certificates interesting and your enthusiasm is contagious." So thank you, she says.
Great. Yeah, you're welcome. Once again, I did see a comment that a lot of agrologists and stuff are trained to read the certificates, and that's probably because they've sat on the phone with someone like me until they figured it out, right? And so if you're new to it, and you see the lab's phone number is on the certificate, you can actually phone the lab in question and ask what they've done and what it means. So yeah.
That's right. Yes. And then along those lines, Sheila had mentioned is information available on your website regarding the crop certificate number?
So probably we don't have information on that, but that's something that we could certainly add. On our website at seedcheck.net, we do have links to some organizations, the Canadian Seed Growers Association, the Canadian Seed Institute. As I mentioned, some of those are combining into Seeds Canada, with the exception of the seed growers. So they are working on a more interactive site, but you could still access their sites at this point till their new site is up. They're separate sites. And so there's a lot of information on how to read a certificate available out there for sure. So.
Okay. Thank you. It looks like we have a question from a listener named Linnea, pardon my pronunciation. She says, I have heard that some native plants are very hard to sow or germinate. If you are doing germination tests, what happens if nothing germinates because of the difficulty of meeting the right conditions? Are those concerns that you have?
They certainly are. They're something that we struggle with all of the time. There's kind of two schools of thought on doing some of these difficult to germinate things. We can go through a lot of steps. We can use dormancy breaking techniques, chemicals like potassium nitrate or gibberellic acid. We can, like on switchgrass, for example, we do a 3-week pre-chill before we even put it in the germinator. So going to a lot of techniques to try and break the dormancy. The other school of thought is we just germinate them at a neutral temperature, either that 20 or that 1525 for 2 to 4 weeks. See how many are going to come up right away, and then take the remaining seeds and do that TZ test and say, okay, so 20% are going to come up in the first month, but another 60 are coming eventually over time, probably after the first winter is needed to stratify those seeds and break the dormancy. So it's always a little bit of guess what's really going to happen in the field, but we do our best. Yeah.
Sounds like it. Speaking of dormancy, Kelly is wondering, are there standardized protocols for applying dormancy breaking strategies such as stratification or scarification?
There are for many species, not for everything, unfortunately. There's always work being done, but some things will have a very specific, like 14-day pre-chill or stratification at 5 degrees Celsius. Some things like that scarification works for it will be recommended in the methods. And so, yeah, there are for some species, but not others. Yeah.
Interesting. And Elaine is wondering, after a seed analysis is completed, for how long would you say the seeds are viable for?
That is a super good question, and I wish I had a crystal ball for that one. We can make some generalizations. Things with high dormancy, like Indian rice grass or green needle grass, or things with hard seededness, like purple prairie clover, could last for years and years and years. They could last for 5, 10, 15 years. Maybe even hundreds of years for some things with really high hard seededness. That tends to be more weed seeds than anything. And other things could drop off in a matter of months. Some of the rough fescues, it's good to have a really current TZ or germination on those, because they can go downhill all of a sudden on you, and it's very, very surprising. And so there's no good answer for that one. Yeah, it's all over. It's all over.
Wow. And so how long does it take you from start to finish for a seed test, and from when you get the seeds and you're able to produce the certificate?
Right. Yeah.
I don't know if it varies by species, I guess.
Yeah, it does vary. Usually that very first step, that purity analysis, we have enough people that are experts now that we can usually get that done the very first day. And once that is done, we can take the pure seeds from that. We can report your contaminants and take the pure seeds from that test and send them on for a TZ or germination test. Most TZs are done the next morning, so you already have an idea of quality and viability the next day. But the germination test can take anywhere from 7 days to 3 months, depending on the species. Yeah.
Wow. So it's quite the endeavor, I guess.
Sure is. Yeah.
It looks like we've got another question that's popped in here, and hopefully I can put it up. There we go. Viability would also, it's a comment, would also be highly dependent of storage conditions, but germination generally decreases annually. So an updated seed certificate will help you adjust your seed mix to maintain your target plant community. Do you have any comments about that?
This is absolutely true. So especially when you have a mixture, you have some species that might have dropped off more than others, and so it is a really good idea. I mean, most seed companies are pretty conscientious, and they will retest One of the things that makes us really busy this time of year is they're testing everything that they have carried over from a previous year to make sure that it's still worthy of sale and has a 2021 germination or TZ on it. And if you have seed left over, that's a very good idea to do as well. Once something's already in a mixture, you can imagine it gets a little more expensive, because we have the purity report should always be true of that seed lot. So the contaminants that are in it are the contaminants that are in it. They don't go away unless you reprocess the seed lot somehow. But the germination and TZ can change over time. So as close as possible to your seeding time. So usually like January, February, March, when we're going to be seeding in, April, May, June, July, and August, you should get those retested. And so we have to separate those species out and redo that germination portion, but we don't have to do the whole purity over again. So.
Okay, interesting. I think that's all the time that we have today. There's another comment here about how informative and enthusiastic your presentation was. So I think that's kind of a good place to end it. So thank you so much, Morgan, for your really enthusiastic. We've had it many times today. You're very passionate about what you do. You're obviously very good at it. So thank you very much for taking the time to present to us today.
Thanks, thanks to the organizers for having me. Yeah.
Awesome. You're welcome. To all of our attendees out there, this was the last session of the day, and a recording of it will be available in the near future. And you can just click on sessions, session 4, and you should be able to find it there. Right now, we have the trade show going on in the exhibit hall, and we also have some discussion groups going on too. So the Skinners, Michael Skinner and John Skinner, will be talking about seeds and more seeds and everything to do with seeds. So if you haven't had your fill of seeds, head over to that discussion group. We also have another one going on about aquatic invasives, and that's by Jerry Geyer with Mel Toppy that'll be moderating. And as always, we have an open discussion room available. So if there's not enough space in those rooms, head over to the open discussion room and you can talk about whatever you want. So thank you, everyone. Have a great rest of your day, and we'll see you in the networking section of Feed Loop. Bye.